Welcome to this explainer. Today we're embarking on a journey that honestly feels like it was ripped straight out of a sci-fi novel, but it's happening right now in a real laboratory. We are looking at an absolute scientific marvel, the creation of the world's first fully synthetic cell. We're going to explore how researchers are painstakingly snapping together the fundamental building blocks of life, entirely from scratch, using nothing but non-living chemical components. It is a completely awe-inspiring breakthrough, but as we're going to see, it's also grounded in a really fascinating, sometimes messy reality.
Okay, let's dive into this, because a massive philosophical question anchors this entire explainer. For centuries, biology has basically been a science of observation and modification, right? We take what nature has already beautifully engineered and we tweak it. But what if you didn't have to start with a living organism? What if, instead, you could just pull chemicals off a shelf, mix them up like ingredients in a recipe, and literally boot up a living, functioning system? It really forces us to ask, at what exact point do non-living chemicals cross the threshold into something we call alive?
Well, enter the University of Minnesota and their groundbreaking creation, the SpudCell. And I want to be clear, this isn't just some clever chemical party trick. The team, which is led by synthetic biologist Kate Adamala, has engineered the very first synthetic cell with a complete cycle. That means this entirely constructed entity can feed, it grows, it successfully replicates its own genome, it divides, and it can even undergo selection and competition across multiple generations. By all functional definitions, it's doing exactly what a natural cell does, but it was built entirely from the ground up.
So the crucial point is this realization from Kate Adamala. For a really long time, there was this underlying assumption in science that life required something elusive, some kind of vital force or magical spark that separated biological matter from mere chemistry. But the spud cell basically shatters that illusion. The team has successfully replicated in chemistry what we used to think was only possible in biology. They've proven that the whole complex set of behaviors of a cell, the growth, the feeding, the replication, are really just a series of programmable chemical reactions.
But before we get completely carried away with visions of, brewing up complex organisms in a beaker, we do need a reality check. The science here is breathtaking, absolutely, but it is also in its absolute infancy. Kate Adamala herself actually calls this construct an incredibly wimpy organism. It's primitive. Right now, it basically does nothing other than eat and occasionally manage to pop out a daughter cell. So let's do a rapid-fire breakdown of this synthetic chassis they've actually built here and see just how fragile this milestone really is.
Just 150 to 200 molecules. That is the entire ingredient list for the spud cell. When you compare that to a natural cell, which has literally billions of molecules, this is incredibly bare bones. It's orders of magnitude simpler than anything nature has ever produced. 12 hours. That's exactly how long it takes for this primitive cell to laboriously push through a single replication cycle just to make one single daughter cell.
And 86 degrees Fahrenheit. If scientists don't meticulously hold the cell at exactly this temperature while constantly hovering over it and tending to it, the whole system just breaks down. It is an incredibly high-maintenance scientific marvel. And this brilliantly illustrates the vast, vast gulf between our best synthetic efforts and nature's own engineering. Think about it. Where a natural E. coli bacterium boasts billions of molecules and effortlessly divides every 30 minutes, the spud cell has under 200 molecules and drags on for 12 grueling hours. But the absolute most critical difference here is protein production. E. coli is this amazing, self-sustaining factory. The spud cell, it can't do that at all. Because it can't produce its own proteins, SpudCell is entirely reliant on the laborious, constant care of its makers. The scientists essentially have to hand-feed it externally on a regular basis just to keep it functioning. It is far, far from self-sufficient. Literally, if the researchers step away from the petri dish, the SpudCell just stops. It totally lacks the biological autonomy that we normally associate with wild, natural life.
Now, what's really interesting about this slide is the profound philosophical optimism it represents. Yes, SpudCell is wimpy. Yes, it needs constant hand-feeding. But as Kate Adamala points out, because they built it, they know the full ingredient list. They know exactly what chemicals and what molecules are in there, down to the exact concentrations. It is a fully defined biological machine. And because it's fully defined, it can be engineered. This isn't some mysterious black box like a natural cell. It's a blank, programmable canvas. But naturally,
This brings us to a heavy and honestly fascinating debate in the scientific community. Drew Endy, who is a bioengineering professor at Stanford, completely acknowledges the monumental achievement of constructing a cell, but he draws a hard line at calling it life. His skepticism really stems from that lack of autonomy, because the SpudCell can't evolve on its own, and because it requires constant external human intervention just to survive its 12-hour division cycle. Can we really call it living biology? It's a profound question that seriously challenges our very definition of existence. Yet other experts take a slightly different view. Synthetic cell researcher John Glass points out that even with all its wimpy limitations, the SpudCell is a monumental leap forward. Building a synthetic cell bottom up, which means starting from non-living chemicals rather than just stripping down something that's already alive, is incredibly difficult. Glass notes that this non-living synthetic cell is much closer to being alive than anything else produced in this specific field. It blurs the line between chemistry and biology in a way we have literally never successfully done before. Let's move to and see how this builds into the future. Why does all of this actually matter to you? Well, because even if SpudCell is just a primitive, wimpy chassis today, it is the absolute foundation for tomorrow. The team really hopes this breakthrough is the spark that ignites the true age of the bioeconomy. By creating a basic, completely understood platform, they're providing an enabling technology that will allow future researchers to truly engineer biology the exact same way we currently engineer computer software. Just look at the massive real-world problems we could eventually solve with this. If we treat these primitive synthetic cells as customizable biological machines, the possibilities are just staggering. We could design programmable biotech chassis tailored for highly specific tasks. Imagine engineering synthetic cells that act as microscopic doctors, navigating through our bodies to deliver highly targeted treatments to fight cancer, all without damaging healthy tissue. We could even deploy engineered synthetic organisms into the environment, specifically programmed with brand new, highly efficient methods for vacuuming carbon right out of the atmosphere. The SpudCell is basically just the 1.0 version of a technology that could completely rewrite our future. Which brings us right back to where we started, but now with an entirely new perspective. Today we have a 200-molecule cell that needs to be delicately hand-fed in an 86-degree lab. It's chemistry doing a really good impression of biology. But as we continue to add to that ingredient list, as these synthetic cells become more complex, more autonomous, and more capable, that line between a chemical machine and a living organism is going to completely vanish. So I'll leave you with this. If we can engineer a basic cell from scratch today, what will we define as living tomorrow? It's a question we are going to have to answer a lot sooner than we think. Thanks for joining me on this explainer, and I'll catch you on the next one.